Thank you for coming to this afternoon's session. This session is Stem Cells and Mental Health, not of the moderator and the presenters, but addressing mental health issues. And as many of you know, dealing with brain diseases have been incredibly intractable over the years. Lack of adequate model systems, screening paradigms, or understanding the basic biology. And we have two of the leaders in this field making major advances speaking today. The first speaker will be Daniel J. Lodge, who's at the UT Health Science Center in San Antonio.
Thanks. I'd like to thank the organisers for inviting me here today, and I'm going to be talking about some preclinical studies we've been doing that suggest that stem cells or cell-based therapies may actually be an effective therapy for psychosis in schizophrenia. So schizophrenia is a debilitating disorder. It affects up to 1% of the population, and it's characterized by three sort of distinct symptom clusters. The first are the positive symptoms. They include things like hallucinations and delusions, and this is going to be the sort of topic of the work that I've been doing, focusing mainly on positive or models of positive symptoms of the disease. But there's also negative and cognitive dysfunction associated with schizophrenia that's not currently treated well with any psychotic drugs, and it's something else that we're looking at from a cell-based therapy as well. So we don't study schizophrenia, we study analogues in a rodent model. The model we use is known as a methylase oxymethanol acetate model, and what it is a developmental disruption model where we take a pregnant female rat at a specific time of gestation and inject her with a mycotoxin known as MAM. What this does is reproduces a number of histological, neurophysiological, and behavioural correlates that are somewhat analogous to some of those seen in human schizophrenia patients. And for the sake of brevity, I'm not going to go into what they are, but I'm more than happy to talk about them. We've published on this quite extensively. So I'm a trained electrophysiologist. What we typically do is record neuronal activity in the brain, and what we're trying to get at is what the primary dysfunction is in schizophrenia with the hope that we can come up with new therapies. This is some old work I did looking at dopamine neuron activity in this model of schizophrenia, and what we saw is that these rats have about a twofold increase in dopamine neuron activity throughout the ventral tegmental area. This is significant because increases in dopamine transmission are pretty much the oldest hypotheses of psychosis in schizophrenia. All patients, or not all patients, but some of the studies looking at patients have all demonstrated increases in dopamine transmission in the brains of patients with schizophrenia. Normal individuals, if you give them dopamine agonists and indirect agonists such as amphetamine or cocaine, you can actually induce a psychosis-like state. And patients with schizophrenia are a lot more sensitive to amphetamines and cocaine. In addition, we know dopamine's involved because if you give a dopamine antagonist, and all current antipsychotic drugs are dopamine antagonists, they quite well treat the positive symptoms of the disease. So we have pretty much a good literature suggesting increases in dopamine transmission underlie psychosis in schizophrenia, but what we don't know is why. And if you look at the dopamine system, there doesn't really appear to be any primary pathology in this system. So this has led us and a lot of other people to think that it's the regulation of the dopamine system that's dysfunctional in schizophrenia. And I'm not going to go into a lot of the background that supports it, but what we think and what we've demonstrated is that the increase in dopamine transmission is directly attributable to an increased activity in another part of the brain, and this is the ventral hippocampus. The way we examine this in our animals, we're doing dopamine neuron recordings. Here's our twofold increase in dopamine transmission in our model of schizophrenia. If we simply shut down the hippocampus, what we can do is bring that right back down to normal levels. So these guys look just like control animals. What's interesting about this is that it's consistent with recent evidence from imaging studies in human patients suggesting that there is hyperactivity in the hippocampus at rest and that it's directly correlated with positive symptoms of the disease. So that's great. We've taken one hypothesis, the dopamine hypothesis, that suggests that there's too much dopamine in the brain, and now we're saying there's too much activity in the hippocampus, but it still doesn't get to a cellular basis for what we think may be happening. So we went to the clinical literature and looked at a wealth of data from post-mortem studies that consistently demonstrate a specific loss of a certain type of inhibitory neuron throughout the cortex and the hippocampus. Our theory was that if you lose function of these inhibitory cells, then that brain region is basically going to go into overdrive. And the cell that's more consistently implicated in schizophrenia is the inhibitory cell that contains a calcium-binding protein known as parvalbumin. So a decrease in parvalbumin, messenger RNA, and cell number is consistently observed post-mortem in patients throughout the entire cortex. It's also been reported in the hippocampus. So we looked in a model of schizophrenia using both immunohistochemistry for cell counts as well as Western blot across a number of different ages, and we consistently see decreases in parvalbumin expression and cell counts throughout the hippocampus and other regions in this model. But this is not, this is correlative. It doesn't really tell you that anything's causation at all. And what we're interested in is whether this loss of interneuron function specifically was sufficient to recapitulate some of the symptoms that we see in our model of schizophrenia. So to get at this, we used a viral approach where we took lentivirus and we made it express short hairpin RNA targeting parvalbumin. So we took this virus, we injected it into the ventral hippocampus of quote-unquote normal animals, and then we looked at hippocampal activity and dopamine neuron activity. Starting first at activity in the hippocampus, what we see is by simply knocking down parvalbumin protein, we recapitulate that hippocampal hyperactivity. We get an increase in firing in the hippocampus. Similarly, by knocking down parvalbumin in the hippocampus, we drive dopamine neuron activity up. So now we have something that we think may be a key pathology in schizophrenia, in that a loss of parvalbumin interneuron function can drive hippocampal hyperactivity and the increase in dopamine neuron transmission. So as electrophysiologists, we like to do a lot of this. We like to record cells in the brain and show graphs that have a 50% increase in activity. But what is really important is whether this translates into a behavioural change in the animal. One of the behavioural correlates that we look at is simply the response to psychomotor stimulants. And I mentioned earlier that schizophrenia patients consistently demonstrate enhanced response to psychomotor stimulants. And we can do this quite simply in our animals by giving small doses of amphetamine and looking at locomotor activity. What we have here is locomotor activity across time. This is a small dose of amphetamine here, and this is a larger dose of amphetamine administered here. And what we consistently see in our animal models is an exaggerated response. Here I'm demonstrating that just simply knocking down parvalbumin in the hippocampus is able to recapitulate that finding, and what we get is a significant increase in the locomotor response to amphetamine in these animals. So this is our hypothesis, is that a loss of a specific type of interneuron in the hippocampus is sufficient to drive hyperactivity in that region, and that hyperactivity is what drives the dopamine hyperfunction that underlies positive symptoms of the disease. This led us to the suggestion that maybe replacing these deficient interneurons may be an effective and ultimately a permanent solution for the treatment of psychosis. So to examine this, we actually did cell transplants obtained from fetal tissue. We took fetal tissue from the medial ganglionic eminence. This is the primary source of developing GABAergic interneurons in the brain. We used green fluorescent protein rats so that as donors, so that we could follow the fate of these neurons to see if they survived, migrate, and to determine the phenotype of these cells. And what we wanted to know is if it could rescue these deficits in GABAergic signaling that we see in our model of schizophrenia. So here is the histology we did post-mortem at the end of all these experiments. This is at least 6 weeks following transplantation. What we see is that these fetal neurons migrate away from the site of injection. They obviously survive. They have processes, and they seem to have integrated within hippocampal circuitry. Most importantly, they also display GABAergic markers so that they have a GABAergic phenotype. So we're specifically transplanting inhibitory cells back into the hippocampus. So what happened, you may notice up here we have two groups. We have some that are called dead and some that are called live. This doesn't refer to the animal, this refers to the cells that we transplanted. Obviously putting cells in the brain produces an immunological response. We wanted to control for that, so we took some cells and killed them, and they were injected for control purposes. So what we have here is activity in the hippocampus. Again, our model has a significant increase in hippocampal activity. We have this hippocampal hyperactivity. If we look in rats that had the MGE transplants, this was completely restored. We then looked at dopamine neuron activity in the VTA, and I've shown this before. We have a twofold increase in dopamine neuron activity in the VTA in our schizophrenic animals. And if we look at rats that had the MGE cells transplanted back into the hippocampus, we get a complete restoration of function. And it's important to note that this comes right back down to control levels. It doesn't go any further. One of the biggest problems we have with current antipsychotic drugs is they shut down the dopamine signal. And the dopamine signal is a key signal for reward, feelings of pleasure, and a whole host of other things. So it doesn't decrease dopamine below levels, it just restores function back in this system. Again, we like to show that this has some behavioural relevance, so it actually changes behaviour in the freely moving animal. And so we, again, looked at the locomotor response to psychomotor stimulants. In the red here, what we have is the exaggerated response to amphetamine in our model of schizophrenia compared to our control animals here in blue. And in the pink, what we have are the rats that had the cell transplants into the ventral hippocampus. And what you can see is that this manipulation completely restored normal function in this behavioural assay. So just quickly, this is a stem cell meeting, and technically what we took out was fetal tissue. These aren't stem cells, these are already interneurons. And one of the other caveats of this is the MGE is a birthplace for a whole host of different types of interneurons, including those that express parvalbumin, but also other types of cells, such as those that express somatostatin. And we really wanted to look at the relative contribution of parvalbumin or somatostatin. Does it have to be a certain type of interneuron that's transplanted back in, or does any neuron basically suffice? So I mentioned we're not molecular biologists, so we collaborated with someone that was, or that is. Stuart Anderson has been working in this field for a long time. He's up at Penn. And we were interested in taking embryonic stem cells and turning them into either somatostatin-enriched or parvalbumin-enriched neurons that we were then going to transplant to the hippocampus and see if they restore function. If you look at sort of the life cycle of these interneurons, they express a couple of really important markers. They start off expressing NKX 2.1, and then once they're postmitotic and migrating, they express LHX6. So we took advantage of this. Stuart Anderson has an embryonic stem cell line that has green fluorescent protein under the control of LHX6 and M-cherry under the control of NKX 2.1. This allows us to separate early and late-born neurons, so we can enrich for parvalbumin and somatostatin. The way we do this is we use FAAPs to basically identify the green and red cells and inject them into the hippocampus to see what that does. So these are preliminary data sort of to demonstrate feasibility to see if this may actually work. What we've done so far is sort of the first cohort of these experiments. We've shown that the cells survive and that they display the phenotypic markers that we expect them to. So these cells were selected for parvalbumin and do indeed display parvalbumin in those neurons. These cells were selected for somatostatin and show co-localization with somatostatin. And we have some really preliminary data showing sort of feasibility of this. Here are our control animals that I've shown previously with our model of schizophrenia showing an increase in dopamine neuron activity. And here's our preliminary studies suggesting that either parvalbumin or somatostatin neurons derived from embryonic stem cells are able to rescue this increase in dopamine neuron activity. So with that, I'd like to conclude and say that we think schizophrenia is associated with an increased hippocampal drive of the VTA. This is not something that we've just examined in a rodent model. This is something that is supported by imaging studies in human patients. We think it's attributable to a decrease in interneuron function, and we've shown that by using lentivirus to specifically knock down this type of interneuron, we can recapitulate some of the symptoms that we think are analogous to those in patients. We've shown that transplantation of these sort of precursors from the medial ganglionic eminence is able to restore aberrant activity within the hippocampus and also downstream in the dopamine system as well. And we have very preliminary data suggesting that cells derived from embryonic stem cells that express either parvalbumin or somatostatin may also be effective in this model. And ultimately down the road, what we hope is that stem cells may actually be a novel therapeutic approach for psychiatric disorders, especially those that have psychosis as a dimension. So with that, I'd like to thank the people that did the work. Stephanie Perez is a grad student that did a lot of the work with the MGE transplants. Jennifer Donegan's a spectacular postdoc that's taken over the embryonic stem cell project and generated that preliminary data. Stuart Anderson is a collaborator at Penn that provided us with not only the cell line but the technique and the expertise to do these studies. I'd like to thank financial support, and I'd like to thank you for listening.
Mary Pat Moyer, NSAL here in San Antonio. How long do these cells persist in those animals, and or have you only done short-term studies?
We haven't done long-term studies.
Yeah, long, like a couple of years or something?
Yeah, we haven't done year-long studies, but we have done studies that are certainly in the 4 or 5, 6 month range, and we still have cells that have seemed to have become somewhat permanent.
And you see no evidence of tumor formation or anything like that?
No evidence at all.
Okay, thank you.
I'm sure you're aware of this, but there's a CERM disease team funded proposal headed up by, I think, Arnold Krigstein at UCSF, proposing to use ES-derived interneurons for the treatment of epilepsy. Mm-hmm. And I just wanted to make sure that the two groups were talking enough, because you can learn so much from each other. Yeah,
I think... So when we came up with this hypothesis a fair few years ago now of hippocampal hyperactivity sort of underlying psychosis, we've taken a lot of the leads that have been done in the field of epilepsy that have been targeting hippocampal hyperactivity for a long time, and so they've sort of taken the lead in this, and I think talking across the different groups is something that's certainly warranted.
Can I ask you one question? You may have addressed this. How do you think the patients come to have this decreased hippocampal input to the VTA?
So we think it's due to the decrease in interneuron function. What causes that? We're starting to do some studies to get at that. So schizophrenia, we know, is a developmental disruption, sort of a dysfunction. And what we think is things such as prenatal inflammation may actually be underlying some of this. You combine that with the genetic susceptibility, maybe early life stress. These cells are really fast firing. They're susceptible to oxidative damage. So I don't think there's one thing that's going to cause damage to an interneuron. I think there's multiple things that can cause damage to these cells, and I think that could be why this is a sort of heterogeneous disorder. It's not one gene, it's not one environmental manipulation that produces the disease, it's a combination of these things.
Right. Do you think it's a degenerative process, or it's a hit and run, and then why is this cyclical, and you have these schizophrenic breaks, but then there can be normal periods?
Yeah, that's a good question. So we think everything sort of is a developmental issue. So you have abnormal migration and abnormal generation of these interneurons. We know that the sort of normal physiology of the system doesn't mature until adolescence, late adolescence, so we think that's why symptoms don't appear until late adolescence. The cyclical nature of that, we think may be due to the cyclical nature of the hippocampal hyperactivity, maybe due to clock genes and things like that, but that's speculative.
Shouldn't be a problem. Am I on? So, as you can tell by my title, that I'm very sympathetic to the developmental aspect that we just heard about before. And I'm going to be talking about another disease, but I also believe that aberrant development probably plays a role in a lot of these neuropsychiatric disorders. And the work I'm going to be talking about, which is still a work in progress, is attributable to the work of a whole team of people. As you can see, there'll be many, many aspects, none of which I could ever do, certainly not do on my own. So anybody who's followed the IPS field knows that we've become very good at being able to model monogenic diseases and other diseases with single gene defects. The real challenge, however, is going to be how to use disease in the dish technology to model polygenic, complex, multifactorial diseases, particularly those where we don't have any idea what the pathway is, what the gene is, what the cells even involved are, and all of that's poorly understood. And I don't need to tell this audience that the poster child for that are neuropsychiatric disorders. So not only are they very important in and of themselves, but being able to be able to tackle those, particularly with IPS cells or any kind of disease modeling, really may help give us insights into how to model other complex disorders. So I'm just going to discuss over my 15 minutes a strategy that has proven useful for us and might be used by other people in their own particular disease, whether it's neuropsychiatric or not. And we call this the molecular can opener strategy. And I'll go through it in detail, but basically the notion is to use a known functional agent, even if you don't know how that agent works in a particular disease context. And in this case, it's going to be lithium in bipolar patients. But that provides a molecular hook, a molecular can opener, so to speak, to pry inside the cell, find the target of what that agent is, and then work backwards. Start with the target and then start fleshing out the pathway of which this is a node, upstream and downstream, and see if you can then flesh out the pathway. Then you can find better drug targets, because as we'll talk about, lithium is no day at the beach for patients who have to take it for bipolar disease. So most psychiatric disorders are difficult for all the reasons that you've heard and that you know about. The diagnosis is often very imprecise. When you do use drugs, the mechanism of action is usually not known. It's rarely specific to a particular disease. The responses are heterogeneous, and most of the time the choice of drugs is purely empiric. There's one exception to this in all of neuropsychiatry, and that's bipolar disease, because it's known that 50% of the patients respond to lithium. Nobody knows how, nobody knows why, nobody knows what the mechanism is. However, it's pathognomonic, in fact. If you have a patient who's responding in a particular way and you start him on lithium and he responds, by definition, he has lithium-responsive bipolar disease. In all of psychiatry, the only other disease that really can be diagnosed this precisely is anorexia nervosa. So, but that became an interesting disease to begin to look at lithium-responsive bipolar disease. Now, I won't have time to go into all of the aspects of bipolar disease except to say a few things. One, it used to be called manic depressive disorder, so that already tells you what the essence of the disease is. It's an episodic mood dysregulation. What I didn't know until I started studying this is just how highly lethal it is. It's an amazingly lethal disease because of suicide, because of drug abuse, and then all the comorbidities that are associated to this. It's exceedingly prevalent. Most of the time it's hidden by families, but it is exceedingly prevalent. And then it runs in families. if there's somebody who has the disease in one family member, then it's usually present in multiple family members across multiple generations. So it's heritable, even though nobody's really been able to figure out exactly what that heritable gene or component is, and it has a developmental field. Even if the patient didn't really present or get the diagnosis until young adulthood or a teenage, you talk to family members and they'll say, I got the sense that there was something wrong or something different straight away, right from the earliest stages of infancy or childhood. So the approach is basically, in this case, is find lithium's protein targets, which means we're going to leapfrog over transcriptomics and go straight to proteomics. And then, as I said, try to reconstruct the lithium response pathway to try to get its mechanism of action, and in that sense, flesh out the entire story and see whether we can get to the heart of bipolar disease. So it's not that lithium action is not known. People, it's known that lithium regulates protein kinases. It plays a role in phosphorylation, but that's just not specific enough, because phosphorylation does all kinds of things. It turns genes on, turns genes off, and it affects many of these. So what these guys in the lab decided to do was to make iPS cells from lithium-responsive bipolar patients. So you have skin biopsy specimens, you turn them into fibroblasts, then you reprogram them to iPS cells, and you get pretty pure populations of neurons. And then what we do is look at the proteome. And the technique that we did was kind of a fairly simple technique, but very, very effective. It has very high stringency. Take one clone of iPS cells and expose it to lithium or not, and then do 2D differential gel electrophoresis. I don't know if you can see it, but what it basically does is look at the spots that change in response to lithium. And it separates them based on molecular weight and charge. And what you're going to look for are not the yellow spots, because those are the same, but the spots that are either just green or just red, because they've been altered in some response. They're different in response to lithium. And then we prayed, and we just prayed that we wouldn't have too many spots. It would be a manageable amount of spots, and we lucked out. There were only, in this particular set of experiments, there were only 15 spots that were different, which meant that they could be picked, identified by mass spec, and take a look at them and see what they were. Now, one candidate that came out that was totally unexpected and had never been implicated before, but as a developmental biologist was incredibly interesting to us, was CRIMP2, and I'll tell you what that is in a second. It stands for collapsin response mediator protein 2. Now, collapsin is the old name for semaphorin 3A. So this starts becoming a little more interesting. Semaphorin is necessary for neurite outgrowth, and semaphorin cannot work without this mediator. That's why it's called the mediator protein. So it's carried on chromosome 8. It's essential. It's pivotal for brain development, and what it's necessary is for neurite outgrowth, axonal guidance, intracellular transport, laminar positioning, the distinction between whether a cell is going to become excitatory or inhibitory, growth cone collapse, neuronal polarity, cilia function. So it's already becoming very important as this molecule it's necessary for ion channel function. Already this starts fulfilling what our desire was, something that's heritable and is pivotal in development. Its function is completely dependent on its phosphorylation state. So that starts becoming interesting as well. It responds to external cues. So obviously we didn't just focus in on that to the exclusion of anything else. We did the bioinformatics and in fact found that if you take all of the 2D Dij molecules and you start doing, I know this looks like the Delta airline flight route, but, and friends of mine call this the radiculome. So you're not going to function, we're not going to stay there, except to say that the most important nodes of all of these start focusing on axonal guidance, which is starting to emerge as a real theme in a lot of psychiatric disorders and a lot of degenerative disorders, and including autistic disorders. So it looked as if we were focusing on the right thing. Now, interestingly, another colleague, completely independently, Steve Haggerty at MGH, also was interested in trying to find lithium's target, and completely independently using SILAC, which is an entirely different proteomic type of method. It's looking at marking the amino acids in a differential way. He came up with CRIMP2 as lithium's target as well. And we just happened to bump into each other at a meeting, and we were giving sequential talks. And just before we went up to the stage, our guys had given our talk, and as we were coming off the stage, he said, you know, we hit the same target. So we knew that we were probably on to something correct. Now, anybody who works in the IPS field knows that you can't just go with one IPS line. Certainly from the same, you can't even go with one IPS line from one patient. You have to do multiple IPS lines from multiple patients and multiple lines from the same patient, and then controls and related controls, other neuropsychiatric disorders, other neurologic disorders. So we did a lot of controls, and the controls become very important. This simply documents that we made valid IPS lines. And then we started looking at CRIMP's phosphorylation state. Because it's not that CRIMP, we didn't discover CRIMP. CRIMP is known. It just is kind of in everybody's attic. Nobody really studies it or focused on it. But there were certain aspects that were known about it. And it was that CRIMP's phosphorylation state is pivotal for its role in mediating the dynamic remodeling of cytoskeleton and cytoarchitecture. So I'll come back to this a lot. Let me just go through this. You don't have to memorize it. I just want to mention that CRIMP2 binds cytoskeletal elements when it's not phosphorylated. When it becomes phosphorylated, it releases them. And then there's neurite retraction. The way it gets phosphorylated is this serine first gets phosphorylated by CDK5. That gets primed. Once this has happened, then GSK3 beta can phosphorylate these other sites, and this then becomes the key site. So this is what GSK3 beta causes this phosphorylation. When it gets phosphorylated, it's inactivated, it releases the cytoskeletal elements. And I'll come back to this a lot. So CRIMP2 doesn't start getting expressed until you start getting neural lineages. It's not expressed early on, and then as you start getting neural progenitor cells and then neurons, now you start seeing CRIMP2 starting to be expressed. We need to determine where is it expressed in the brain. And it's expressed in the areas that you would expect in a neuropsychiatric disorder. You just heard it's expressed in the hippocampus. It's also expressed in neurons, primarily in neurons, in other areas of the brain, in the cortex, in the cerebellum, and as I said, importantly, in the hippocampus. If you knock out CRIMP2, you get a completely chaotic, disorganized dendritic pattern, as you can see over here, and as you can see over here is this aberrant dendritic pattern. Here's the normal dendritic pattern. If you look at the dendrites that you actually see, here's normal. You can see that there's decreased dendritic spine density, and the spines that you do get, that's quantified over here, are these aberrant spines called mushroom spines. What's interesting is that just recently, Joe Coyle and his colleagues at McLean's started looking at postmortem specimens of actual patients with bipolar disease, and sure enough, that's exactly what they're starting to see in the actual patients, decreased dendritic spine density and some of these abnormal spines. Now, given that phosphorylation is really important, how is CRIMP2 distributed? Because this might be a key as well. Well, the non-phosphorylated active form is present, and I'll show you some data to this, is all throughout the neuron. When it gets phosphorylated, it then either is excluded from the spine or retreats from the spine. It's just not present in the spine. And here's what you can see over here. So crimp, an active form out in the spine, when it gets phosphorylated, excluded from the spine. And that's simply what a lot of this begins to show, as you can see over here. Which then might explain what we saw, that if we looked at the calcium activity, which is a poor man's way of looking at electrophysiology of neurons from lithium responsive and non-lithium responsive and then unaffected patients, interestingly, the lithium non-responsive, the lithium responsive patients have calcium activity of a completely different slope than you would see of the non-lithium responsive and then wild-type patients. This kind of pattern goes along with what you would expect with dendritic spine abnormalities. It also suggests that it's okay for us to use what the clinicians, some of the distinctions that the clinicians make, which is lithium responsive or non-lithium responsive, may be a valid way for us to continue carrying along in our studies, as you can see over here. So we began to formulate this hypothesis of what we thought might be going on, that CRIMP2 binds these cytoskeletal elements, GSK3 beta phosphorylates it, and you get this CRIMP2, phosphorylated CRIMP2. What we think might be going on, however, is that the set point for bipolar patients is just too high. And I'll show you some data that might support that kind of hypothesis. What lithium would do is then inhibit this and probably also inhibit this and then just lower the level to a good baseline level. And that's what our speculation is. So we began to play out this hypothesis. If you just do staining of IPS of neurons, not only from lithium responsive IPS patient, bipolar patients, but from unaffected patients and even ES cells, you can see that lithium in everything will lower the amount of phosphocrimp. It does it in everybody. But the starting level in these patients already looks, as you can see in immunocytochemistry, to be a bit high. So now we need to start mapping the lithium response pathway to and from CRIMP2. So again, here's the story. CRIMP2 binds cytoskeletal elements like tubulin. It gets primed by CDK5, so that gets phosphorylated over here. And then GSK3 beta puts on the other phosphate groups. The other phosphate groups, then it releases tubulin. So now it's no longer bound to cytoskeletal elements. So part of the pathway should be lithium inhibiting this. And GSK3 beta gets inhibited by getting phosphorylated. So that, so far, so good. Sure. So with that, GSK3 beta inhibition alone should lower phosphocrimp2 positive neurons. And that's exactly what you see. So a diminution as well, in the same way that lithium did it, GSK3 beta inhibition alone also starts lowering phosphocrimp positive neurons. So here's and now here's the Westerns. Lithium does it. GSK3 beta also gets rid of it, and it does it in a dose-response manner, as you can see over here. So here again, lithium increases the inactive form of GSK3 beta, as you can see over here. That results in lowering of inactive phosphocrimp2. Whether you do this through lithium indirectly, lowering and increasing this inactive GSK3 beta, or you do it directly by GSK3 beta inhibitor, and you lower phosphocrimp2, what that then does is increase the association, as you can see in these pull down, of CRIMP2 with these cytoskeletal elements. So now lithium also increases AKT, which means now lithium can not only inactivate GSK3 beta directly, it can do it indirectly. So this is another part of the pathway, as you can see over here. Now, lithium will also decrease this phosphorylation. Remember, this is the primer, serine 522, but it seems to do it independently of CDK5. So now, here's the other part of the pathway. Lithium seems to be doing things independently of this pathway, directly right on CRIMP2 over here. And then there's a balance. There's not only the kinases, there's the phosphatases that remove phosphate groups that should balance that. So looking at over here, lithium also increases the complex that's necessary for these to work. So here's the other part of the pathway. And then lithium probably, we think, works directly over here in ways that need to be worked out. So now we're starting to have the lithium response pathway. Is this disease specific? And remember, the hypothesis is that the bipolar patients already have too high of a set point. And on the Western, that seems to be the case. The bipolar seems to be a little bit too high, but then lithium lowers it, but it lowers it to the unaffected level. And of course, like I said, lithium works on everybody, but it takes the high set point and decreases it over here. Other diseases simply don't have that, like a major depression, and this is related to this guy. His level is already just at the unaffected level. Here you can see it again now looking at GSK3 beta inhibition. Bipolar patients, the set level is just too high, but GSK3 inhibition lowers it to the unaffected level and maybe even a little bit lower, just the same way. So if you start looking at lots of patients now, the set point for lithium responsive bipolar patients just seems to be lower, but lithium then lowers it to the normal set point that you would want to expect. So one way to start looking at this is that you have a higher than normal level of phosphocrimp. Well, at its extreme, and this affects dendritic spine function. At its extreme, you have abnormal spine function, which then gives you, the extreme is just knocking out CRIMP2 and you have no activity at all. So you have what you would see in the mouse, kind of no or diminished dendritic spine function. It seems to be emulated, as we can see, in the specimens that I show you from the actual patients, and in the actual patients in both Burnham specimens, they do also seem to have abnormal crimp handling, particularly phosphocrimp handling. Is it drug specific? Well, only lithium of all the drugs, including those that are used in non-lithium responsive patients, will lower phosphocrimp. And only lithium seems to increase the inactive form of GSK3 beta. Can this start becoming a biomarker? Well, lithium responsive patients, yeah, lithium lowers the phosphocrimp. Lithium non-responsive patients, either their level, it doesn't do anything. Either because their level just is not that high, or it simply seems to be unresponsive to lithium, really unresponsive. Now, that could be that it just starts excessively high, so you lower it and you just never get it low enough. But that's not what the data indicates. It could be that the phosphocrimp is somehow in those patients is just resistant to lithium. But I think what's more likely is that non-lithium responsive bipolar disease is just a different disease. It phenocopies bipolar disease, but it's a different disease and is not tethered to the lithium response pathway, which is this fundamental, whoops, don't want you to know my schedule. So I'm going to wind up now, but I showed you a lot of in vivo stuff. I mean, a lot of in vitro stuff. The key, however, is going to be, does this have any in vivo or functional relevance whatsoever, particularly focusing on CRIMP2? Well, if you give lithium to mice, it increases the inactive form. It inactivates GSK3 beta. It also lowers phosphocrimp. Now, there's very bad models in mice, in animals, for neuropsychiatric disorders. And you heard one for schizophrenia. It's not ideal. Same thing with bipolar disease. But it's what we have. It's what we have to live with. And giving methamphetamine is one of the ways to mimic some aspects of bipolar disease. If you give methamphetamine to mice, first, if you give methamphetamine to the cells in a dish, it increases phosphocrimp. And in the animals, it increases phosphocrimp. Now, one of the behavioral assays of this that's used is looking at manic behavior in response to methamphetamine. So a normal animal in the open field over here spends lots and lots of time exploring the unprotected center. That's a non-manic, low anxiety animal. You give them methamphetamine or they have other kinds of abnormalities, they spend very, very little time in the center, and instead they circle, and spend all their time out here in the periphery, just madly circling the periphery over here. We can quantify that. But now we decided what we would do is do this experiment in an animal whose CRIMP2 is incapable of being phosphorylated. And we can do that by knocking out or knocking in this primer. Get rid of serine 522 so it can't get phosphorylated, so now the whole CRIMP2 cannot be phosphorylated. It would be like an animal like this is like he's chronically on lithium. So now you have the wild-type animal and this animal. He's in the cage. They're doing fine. They're in the center. You give them methamphetamine. The wild-type animal starts acting manic. He spends very little time in the center, circling. But the CRIMP2, the non-phosphorylatable CRIMP2 animal, it's as if he's on chronic lithium. Just nothing changes. Stays right there in the center, and that's quantified over here. So it looks as if maybe we've done some mapping of an important developmental pathway, the lithium response pathway, that might be central to at least some of the aspects of lithium-responsive bipolar disease, as you can see here. What we're trying to do is not only try to understand what developmental role this plays and what inheritable role it plays, but all the alternative ways of lowering phosphocrimp2 instead of lithium. Because lithium, anybody who's known anybody on lithium or has taken lithium, like I said, it's no day at the beach. But maybe there's better ways to get to this, including doing repurposing of drugs that are already FDA approved that can do that. And we think we found some cancer drugs that may be able to do that. The other thing is to speculate whether once we have this and we recognize that this is kind of pivotal over here, this fact that releasing these guys, that this form of CRIMP2 also causes these guys to dimerize and precipitate out. Could something that lowers phosphocrimp2 also be widely therapeutic for other things? And this is something that we're exploring. Maybe this could be the source of other tauopathies, like chronic traumatic tauopathy in response to head trauma and concussion and things of that sort. So I'm going to end by saying that we've generated iPS cells from lithium-responsive bipolar patients, which will yield electrophysiologically active neurons, that using lithium, even though we don't know why lithium works, but we used it as a can opener, so to speak, to get inside the pathways, to find its target within a disease that heretofore is poorly understood. And we did it not by looking at the transcriptome, but by looking at the proteome, and found an unanticipated lithium target, which we think may give rise to the lithium response pathway, which would suggest that bipolar disease is a developmental disorder and can explain some of the manifestations that we see based on this developmental aberration. That maybe excessive forms of CRIMP2 that is responsive to external cues and things of that sort may explain some of what we see in these bipolar patients, that lithium kind of reduces this overly high set point. And it seems that we have some support in mutated mouse models and in human specimens to suggest that maybe we are on the right track, that maybe non-lithium responsive bipolar disease is really a completely separate disease, and we've seen lots of that in medicine. And then I'd just like to leave you with the suggestion that maybe this molecular can opener approach can be used by other investigators with other can openers for other diseases. The key is that you have a black box, essentially, and the iPS cells are modeling that. The black box is being a disease that you don't know much about. All you have is the iPS cells, but your handle for prying into the cell to getting is, and bipolar was, the poster child for this, and we use lithium. It could be Rett syndrome and IGF. It could be whatever you want, but you find the target and then you work backwards, upstream and downstream. You flesh out the pathway, and then you not only try to understand what it's doing, but find alternative drug targets. So thank you very much for your patience.
So there's a lot of neurological conditions where the cytoskeleton gets altered with the same protein.
It's this kind of thing is, like I said, this protein is not unknown. However, there's probably only about 3 labs that study it. And you go to everybody who focuses on the cool aspects of cytoskeleton. And I'll talk to them now, and I'll mention this, and they'll go, oh yeah, that was somewhere in my data set, but we didn't know what to do with it, so we just put it's up in the attic somewhere. So now people are like going in and pulling out these data about these various things. And I think I'm not alone in starting to think that cytoskeletal abnormalities in function and everything it does is starting to play a more and more important role in many different kinds of diseases. When you think of all the things that cytoskeleton does, it's ion channel function, and not just connectivity, but also external transport. So I think that is where a lot of our work in neurodegenerative diseases is going to be going. And it has. And so when you're stuck with something like that, you go, well, boy, that's a really ubiquitous molecule, or you have to try to figure out some way to make it make sense. And if you go, well, what it's doing is it does many things, obviously, but this might be why we see it in so many different things. This is the pathway, and it's not just CRIM2. I mean, it's this whole pathway to controlling cytoskeletal dynamics that is being altered.
Evan, it's really nice work. Beautiful. I have 2 related questions. One is there any work on why lithium chloride works to extend the clinical course of the patients with ALS that may be related to this, obviously acting down lower? And that raises the other question. I mean, there was hand-waving about those studies that it was an antioxidant. So when you focus down on the proteome, we may be forgetting other things. So if you looked at pH changes, redox changes, antioxidant, anti-inflammatory effects, sort of general effects as well of lithium.
General effects on what lithium is doing and things like that? Yeah, well, the one good thing about this technique is that it's very stringent in that it integrates, it takes all these things into account and gives us these targets. Now, some of the other aspects of the 2D DIGE that came out, the other 14, we could, like a jigsaw puzzle, figure out where they went into the pathway, and some of them do change metabolic parameters there. The good thing was that when you did the bioinformatics, the node onto which they were all upstream of happened to be this particular molecule. So I think it absolutely is playing a role. And all we had to do is it was a filter. We wanted a final common integrator of all of those things that might have been changed. But you're completely correct, sure.
Thank you.